Hi everyone, I'm Madam Tawa here. This week's lesson is actually continuation from previous lesson. So we are still in chapter 2, Cellular Respiration. At the end of this lesson, you should be able to explain the biochemical processes in cellular respiration such as glycolysis and Krebs cycle. Let me do a small recap about the previous lesson before you learn about this week's lesson. So in previous lesson, you learned about the basic principles of cellular respiration and also the structure and function of ATP. So this is the overview of cellular respiration. So there are 3 stages in cellular respiration such as glycolysis, citric acid cycle or Krebs cycle, and oxidative phosphorylation. Generally, cellular respiration means is a metabolic pathway in which the glucose broken down to release the ATP energy. Since the cell needs oxygen, cellular respiration is a aerobic respiration. Glycolysis occurs inside the cytoplasm. However, citric acid cycle or Krebs cycle and oxidative phosphorylation takes place inside the mitochondria. Citric acid cycle or Krebs cycle occurs inside the mitochondrial matrix. However, oxidative phosphorylation occurs in inner mitochondrial membrane. Glycolysis means breakdown of sugar or glucose. Glucose is a 6-carbon sugar which breaks down into pyruvate, which is 3-carbon sugar. There are total of 10 steps in glycolysis. Each of the steps also catalyzed by specific enzymes. As I mentioned in previous slide, glycolysis occurs in cytoplasm, and then glycolysis can be classified into 2 major phases such as energy investment phase and energy payoff phase. Energy investment phase starts from step 1 until 5. Energy payoff phase starts from step 6 until step 10. Glycolysis can occur with or without presence of oxygen. So this is the overview of glycolysis. First, glucose breaks down into pyruvate. And then if there is oxygen present, pyruvate will enter the mitochondria to continue the Krebs cycle and oxidative phosphorylation. If let's say there is no oxygen, so the pyruvate will undergo the anaerobic respiration such as lactic acid fermentation and alcoholic fermentation. As I explained earlier, glycolysis can be classified into 2 major phases such as energy investment phase and energy payoff phase. During energy investment phase, glucose uses ATP energy. However, during energy payoff phase, ATP energy produced. So let's look at the first step in investment phase. In first step, glucose molecule phosphorylate by addition of phosphate group from ATP to become glucose 6-phosphate. So this reaction catalyzed by hexokinase enzyme. So how the glucose molecule undergo the phosphorylation process? First, the ATP undergo hydrolysis to produce ADP and inorganic phosphate. So the inorganic phosphate phosphorylate the glucose to become glucose 6-phosphate. So from the structure here, you can notice that there is one phosphate group in glucose 6-phosphate. Then the glucose 6-phosphate rearrange and convert to produce its isomer fructose 6-phosphate. So this reaction catalyzed by phosphoglucoisomerase enzyme. Then the fructose 6-phosphate phosphorylate by addition of phosphate group from ATP to become fructose 1,6-bisphosphate. So this reaction catalyzed by phosphofructokinase enzyme. Fructose 1,6-bisphosphate is actually unstable 6-carbon sugar. So fructose 1,6-bisphosphate split to produce two molecules of 3-carbon sugar, such as dihydroxyacetone phosphate and glyceraldehyde 3-phosphate or G3P. So this reaction catalyzed by aldolase enzyme. Dihydroxyacetone phosphate and G3P are isomers. So dihydroxyacetone phosphate must be converted into its isomer G3P. So at the end of step 5, there are two molecules of G3P produced. This reaction catalyzed by isomerase enzyme. So keep in mind that after step 5, it means starting from step 6 until step 10, you should double everything, since there are two molecules of G3P produced at the end of step 5. So two molecules of G3P undergo oxidation and phosphorylation to become 1,3-bisphosphoglycerate. So this reaction catalyzed by triose phosphate dehydrogenase enzyme. So during oxidation process, G3P donate electrons to NAD positive. So NAD positive reduced to become NADH. So in this step, two molecules of NADH produce. So after oxidation, G3P phosphorylate to become 1,3-bisphosphoglycerate. Then 1,3-bisphosphoglycerate undergo dephosphorylation to become 3-phosphoglycerate. So this reaction catalyzed by phosphoglycerokinase. During dephosphorylation process, phosphate group from 1,3-bisphosphoglycerate removed and transferred to ADP. So ADP here phosphorylate to become ATP. So at this step, there are two molecules of ATP produced. Then 3-phosphoglycerate rearrange and convert to produce its isomer 2-phosphoglycerate. So this reaction catalyzed by phosphoglyceromutase enzyme. Then water molecule removed from 2-phosphoglycerate to produce phosphoenol pyruvate. So this reaction catalyzed by enolase enzyme. Then phosphoenol pyruvate undergo dephosphorylation to become pyruvate or pyruvic acid. So this reaction catalyzed by pyruvate kinase. So during dephosphorylation, phosphate group from phosphoenol pyruvate removed and transferred to ADP. So ADP here undergo phosphorylation to become ATP. So at this step also another ATP energy produced. So total of 4 ATP energy produced during energy payoff phase. And during investment phase, there are 2 ATP energy used. So this is the overview of glycolysis pathway. As a summary, glycolysis occurs in the cytoplasm and can occur without or with presence of oxygen. So from one glucose molecule, there are two molecules of pyruvate produced, can yield 2 ATP and 2 NADH. During the investment phase, 2 ATP used. However, during energy payoff phase, there are 4 ATP produced. So the net yield of ATP is 2 ATP. Then the NADH that they produce will enter the electron transport chain. So from one glucose molecule, there are two molecules of pyruvate produced during glycolysis process. So the pyruvate then enter the mitochondria to continue the Krebs cycle or citric acid cycle. So the pyruvate enters the mitochondria with the presence of oxygen, and the pyruvate must be converted to acetyl-CoA, then only can enter the Krebs cycle. So this reaction known as link reaction. So keep in mind that pyruvate cannot directly enter the Krebs cycle. Pyruvate must be converted to acetyl-CoA, then only can enter the Krebs cycle. This reaction known as link reaction, since it links the glycolysis to the Krebs cycle or citric acid cycle. From glycolysis pathway, one glucose molecule produces two molecules of pyruvate. So pyruvate with the presence of oxygen enter the mitochondria to enter the Krebs cycle. However, keep in mind that pyruvate cannot directly enter the Krebs cycle. Pyruvate must be converted to acetyl-CoA, then only can enter the Krebs cycle. So there are three reactions occur inside the mitochondria during link reaction. First, pyruvate undergo decarboxylation process by removal of carbon dioxide and then undergo oxidation process by donating the electrons to NAD positive to become acetate. Then coenzyme A added to acetate by weak bond to become as acetyl-CoA. Then acetyl-CoA can enter the Krebs cycle. So, since two pyruvate enter, there are two molecules of acetyl-CoA produced. Therefore, Krebs cycle needs to undergo twice. So this is the overview of Krebs cycle. Krebs cycle named after Hans Krebs, who discovered this pathway. So the acetyl-CoA that produced during the link reaction will enter the Krebs cycle. Krebs cycle also known as citric acid cycle or tricarboxylic acid cycle. So generally, Krebs cycle is a series of enzymatic reaction that takes place in mitochondrial matrix. Most of the reactions are oxidation or reduction reaction and decarboxylation reaction. This is the Krebs cycle, which involves 8 steps. First, the acetyl-CoA that produced during link reaction enter the Krebs cycle. Acetyl-CoA is a 2-carbon compound. So the CoA from acetyl-CoA release and then acetyl combined with 4-carbon compound oxaloacetate to become citrate. So this step catalyzed by citrate synthase enzyme. Then citrate is a 6-carbon compound which rearrange by removal of water and addition of water to become its isomer isocitrate. So this step catalyzed by aconitase enzyme. Isocitrate also 6-carbon compound which undergo decarboxylation and oxidation process to produce alpha-ketoglutarate. So this step catalyzed by isocitrate dehydrogenase enzyme. Isocitrate undergo decarboxylation by removal of carbon dioxide and then undergo oxidation process by donating the electrons to NAD positive. So NAD positive reduced to become NADH. So at this step, NADH produced. So this step catalyzed by isocitrate dehydrogenase enzyme. Then alpha-ketoglutarate is a 5-carbon compound, undergo decarboxylation and oxidation process to produce succinyl-CoA. Alpha-ketoglutarate undergo decarboxylation process by removal of carbon dioxide and then undergo oxidation process by donating the electrons to NAD positive. Then NAD positive reduced to become NADH. So at this step, NADH produced. Then succinyl produced. So succinyl combined with coenzyme A to become succinyl-CoA. This step catalyzed by alpha-ketoglutarate dehydrogenase enzyme. Then succinyl-CoA is a 4-carbon compound. CoA attached to succinyl by unstable chemical bond. So, CoA released and displaced by phosphate group. Then the phosphate group transferred to GDP. GDP phosphorylate to become GTP. Then the phosphate group from GTP transferred to ADP. So ADP phosphorylate to become ATP. So at this step, ATP produced by substrate level phosphorylation. Then succinate produced. So this step catalyzed by succinyl-CoA synthetase enzyme. Succinate, also 4-carbon compound, undergo oxidation process by donating the electrons to FAD to become fumarate. So this step catalyzed by succinate dehydrogenase enzyme. Then FAD accept the electron from succinate, reduced to become FADH2. So at this step, FADH2 produced. Then fumarate undergo hydration process by addition of water molecule to produce malate. So this step catalyzed by fumarase enzyme. Fumarate also 4-carbon compound. Then malate undergo oxidation process by donating electron to NAD positive to become oxaloacetate compound. So the NAD positive accept the electron from malate, reduced to become NADH. So at this step, another NADH produced. This step catalyzed by malate dehydrogenase. The cycle turns twice since one glucose molecule produces 2 pyruvate. So as a summary, Krebs cycle is an aerobic respiration since requires oxygen. So as I mentioned in previous slide, since one glucose molecule produces 2 pyruvate and then the pyruvate converted into acetyl-CoA. So 2 acetyl-CoA enter the Krebs cycle. So if each turn of Krebs cycle produce 3 NADH, 1 FADH2, 2 carbon dioxide and 1 ATP, then 2 cycles will produce 6 NADH, 2 FADH2, 4 carbon dioxide and 2 ATP. So the ATP in Krebs cycle produced by substrate level phosphorylation. So when you compare glycolysis and Krebs cycle, glycolysis can occur with or without presence of oxygen. However, Krebs cycle need oxygen. So this is the basic difference between glycolysis and Krebs cycle. Then the NADH and FADH2 that produced during Krebs cycle enter the electron transport chain. So in next lesson, we will continue the third stage of cellular respiration, that is oxidative phosphorylation. So that's all for this week lesson. Thank you.